Well, we're on our way to looking at gene expression, and we've already looked at the transcriptional process, which is actually taking the information off of the gene. Now we want to work our way down to actually eventually making protein, which of course is phenotype. So just to remind us where we are, we talked about some post transcriptional modifications or sometimes co-transcriptional modifications that can occur. Certainly the 5' cap, which protects the first part of the end, is usually co-transcriptional. Then we have a poly A tail, which occurs post-transcription, which involves not only slicing up some parts of the message off itself near the poly A site, but also adding these adenine residues. And again, the length of those will determine how long this message will persist. And then also, of course, in eukaryotic systems, we have to go from heterogeneous RNA to messenger or mature RNA, which involves splicing. There's mechanisms for that, but kind of as a general model, we talk about the spliceosomes, this idea that some RNA and proteins come together and they find these splice sites within these exons and introns, and they bring the exons together and remove the exons, the introns with bilariate structures where they're degraded and reused. So now what we've done is we've matured the message, right? At least in eukaryotic systems, we have this message that can be signaled to be brought out of the nuclear region into the cytoplasm, and we can start to work with it. Now, obviously, what we're going to do is take that gene. We've now made this mature message, and we have worked through the process of transcription. And what we want to do in these few lectures is work through the process of translation. And there used to be, as we talked about in Central Dogma, there used to be this idea they went from gene to RNA to protein. Now we know by alternate splicing, you don't even go from one gene to one RNA, that there can be multiple routes to various types of RNAs by which are the exons that you actually use. And certainly you don't go directly from RNA to protein. It turns out that we are gonna read the RNA by translation and produce a polypeptide, but that polypeptide at that moment will not be a functional protein. It may not be until we combine it with other polypeptides, but we'll talk about actually making the protein once we make the polypeptide first. So let's look at some of the major things about translation. We'll talk about where it happens. We want to talk about what we know about what's called a coding dictionary and work through a model of how we might use it. Okay? So we're looking to go from RNA to a polypeptide chain. It's going to happen in the cytoplasm. We're in this course, we're dealing with eukaryotes, so we need to get it out of the nuclear region into the cytoplasm. And it will be associated with these things called ribosomes, which are generally found along the endoplasmic reticulum. Okay? The primary time that we're going to see it happen is in G1, because this is the period of the cell when the cell's in its normal state. Remember, our DNA is out in its balls on a string, or 11 nanometer fibers, or maybe 30 nanometer cores, but the DNA is available. There's also some time during G2 after replication when you need to synthesize information for the meiotic or mitotic process. And so there's proteins being made then, but that's a much less active time, because remember, as we go through synthesis, we also tend to start the compression process of our DNA into chromosomes. Okay, so why do they occur mostly there? For that very reason. Most of the other time in mitosis or in S phase, you're either you're not replicating DNA here and compressing it, and then of course in mitosis you're certainly compressing it early on and then pulling those sister chromatids apart, so that you don't need to have, or can't really have, because of all the compression, any real transcription and translation going on for the most part. Okay. Now there is some translation that can happen here. Primarily what happens there is that messages are already produced before you get into any one of these phases, and there's simply translations then is allowed occurred, right? Because all the activities going on inside the nucleus is relative to replication or to mitosis. So if there are messages out there, they can be translated, but there's very little transcription. So when it is happening, particularly out here in G1, how is it happening? Well, I think we all know that the way we read the words of a message is through these structures called codons. Very simple. Unlike many languages which have words of varying length, codons only have one length, which is 3 letters at a time. So 3 nucleotides in the message will be considered one codon. One codon is read as a word that correlates with one amino acid. And I just gave some examples. Here's AUG, here's the second. So we just start reading along, and then we would have put the appropriate amino acid for that code, so related form. And it could be whatever forms that we might have. Each one of these is a different sentence if you think about it. And the sentences are very straightforward. They read from left to right as groups of 3 letters. Once it starts, it's a blind process. If it starts in some area, now this is, and they tend to do this in books and places, indicate this is a naked 5' end, but of course we know it has a cap. In fact, there's quite a few sequences out here, quite a few pieces of RNA nucleotides out here before you even get to the start codon. So in many systems, but once you hit the start codons, once that system is set, then you read in the next 3 just in blind sequence until you hit a stop system. Okay. Now the question becomes, how do I know what that word means, right? How do I know what the word UGG means? Well, obviously that's going to require some kind of Rosetta Stone or something else. We call it the coding dictionary for us. It's a very simple dictionary, but it has some important features that I think everyone should know if you come out of genetics. So let's talk a little bit about the features of the genetic code as we understand them. First, it is written in a linear form, just as I said. We read 3, 3, 3, just straight across, okay? We've already said this. It's coded, the words are in triplets, so they're 3 letters, which makes up a codon. Now, the next 2 are words that people tend to confuse. So pay attention to these just for a moment, or look them up in your book and make sure you understand the difference, okay? First term is unambiguous, right? What that means is a codon will give a specific amino acid and only that amino acid. So if you see UUU or you see CUA, you're only going to get one amino acid from that codon, okay? There's no question about it, which means if I know the DNA code, I can know the message. And once I know the message, I can guarantee you that I know what amino acid sequence is going to be put in that polypeptide. Okay, that's unambiguous. That's completely counter to the next term, which is degenerate. Degenerate is the fact that while one codon will only code for one amino acid, there are lots of different codons for the same amino acid. Okay, so you may have this codon may be CCC or CCU, and both of those may be code for exactly the same amino acid. But you know specifically what CCC codes for, but you don't necessarily know which. If you just know the amino acid, you don't know which codon produced it. So it's called degenerate, which means you can come from the top, from DNA to RNA to polypeptide amino acid sequence without any confusion. But if you have the amino acid sequence itself, use proline and serine and whatever, you don't know exactly what the genetic code was just because you know that polypeptide sequence. And we can sequence polypeptides very much like we do DNA sequencing. Different technique, but we can still figure out whether it is, in fact, isoleucine and then serine, guanine, whatever it is, okay? But it's degenerate. Just because I know that code, I can't tell you the exact piece of DNA code that it came from, right? There are very specific start and stops. We're going to concentrate on 4. AUG will be our start code that we'll work from. And then there are traditionally 3 stop codes, UAA, UGA, and UAG. There are some others that can be found around in other systems, particularly the mitochondria has some other variants of these. But for us, these will be the ones that will work. So if you need to know those. If you see a piece of code somewhere, one of the problems that we often give you is you have to figure out which is the 5' end and work whichever direction you need to go and look for AUG and then look for stop code, particularly when we get the mutations, because sometimes what will happen is that you'll have a UUA mutate to a UGA, and of course that'll make a stop code where it shouldn't be. So you have to look for those. So you need to know these 4. Okay. A couple other things. One, it's considered commaless. It means that it's read as 3, 3, 3, 3, 3, 3. It doesn't start and stop or do anything else. In the same way, the code doesn't overlap. We don't read AUG and then pick up the G as the next part of the codon. It's strictly 3 and then 3 and then 3. We don't overlap. We don't use this one as part of the next codon. Okay, so it's non-overlapping. And then the final term is called universal, and notice I have a little approximately sign for it. It's certainly on this planet with just DNA. You can use that code for almost anything. Again, mitochondria will be slightly different, but it's only in a very, very few places. And there's a few other organisms that have some variants of it. But for our purposes, 99.99% of the code, and certainly anything you work in problems, you'll be able to use a coding dictionary. So what does a standard coding dictionary look like? Well, they come in many forms, but this is one that's very common, very classically used. It looks something like this. It just has U, C, A, G set out 3 times, one down this direction, one across the top, and one in kind of small groups like this. To read the code, you simply take the codon that you want to do, which is AUG in this case. You take the first position and you look wherever in this column has the first position, and that's where we're going to go. So A is the first position. It's found here. Okay. So our amino acid that's going to be coded for by this codon falls somewhere in this sector right here. Okay. It's either isoleucine, threonine, lysine, serine, arginine, or something. Okay. It's one of those, our met. Right. Then we go to the second position, which is a U, and we read across the top for the second position, and we just sort of pick that column. Well, that turns out to be the first column here, right? So we are in that column somewhere, which means where they intersect, we're in here somewhere. We're going to find out that AU always codes for either isoleucine or methionine, one of the two, okay? So we're going to be one of those two places. What will determine where we are in those places is the third position, okay? Third position, then we read over where it says third position over here, and we look for U, no, C, no, A, G. Okay, so we want G right there. So now we're going to come back and we're going to be in this little group right here, and obviously that's going to be net. Okay, so AUG codes for methionine as it would. It's the start codon, and that's the first amino acid that's put down just any polypeptide. That's often cleaved off, but that's the first one that's put there during the translational process. Okay. All right, well, there's some other things you need to look at on this thing. You notice that serine's here a lot, leucine is here a lot, and what you have in these is serine is the SER is a classic abbreviation for the amino acid, but to make it simple, like in computer programs and things, we also have little letters. So serine is an S, okay? Cysteine is a C, arginine is an R, because you've already used the A somewhere else, so you have to for alanine, so you have to use something else. But these little letters stand for the same thing, okay? And notice some oddities here. Here's serine right here, and then here's serine way off down here, okay? Which indicates that it can have multiple codons that code for serine that are very different in form, okay? This one would be read as what? A, and then G, and then U. So A, G, U would give you serine, as well as ACU. Okay, so that would actually be a second positional difference that would change in that form, which is very, very rare. Okay, all right. Now, what you see most often the time is not that second position. In fact, it's extremely rare. If you have a second positional change in a codon, it will almost always change the amino acid. Okay, but what won't change the amino acid all the time is what's called wobble or third position changes. Okay, there'll be third positional changes. So let's just take an example. Here's serine up here, right? Just as I said, it doesn't matter whether it's UCU, UCC, UCA, or UCG. They'll all produce serine. Okay, just ignore that it also goes off over here, right? Others that you see that in, I mean, leucine is UUA, UUG, or CUU, CUG. So it's usually third position, like proline right here. All of those are third position. We've got alanine here, valine, they're all third position, right? Now, anything you put in the third position, once you get to GU, it doesn't matter what the third position is, it's going to be valine, right? That's called wobble, third position wobble. But as you can see, there's also some first position wobbles, all right? UUA, right? And CUA both produce leucine, which means the first position can change, but that's much more rare. Third position, very common, called wobble. Second position, almost never going to change. Well, it will change, but it will always produce an amino acid difference if it does change. First position can change a few times, okay? So there's some things that work, right? Methionine is your start codon. Term right here means there is no amino acid put down, right? That's what those, so if we said, what would you say was one? UGA. U, G, and A is a term code, right? UAA is a term code. That means there is no amino acid that would stop the translational process, okay? All right, so people kind of ask this kind of fun question, why is there wobble? Why don't we have all this, right? Well, That's a pretty good question, and I think one answer that's been brought forth by people just thinking about it from a statistical standpoint is this: if we have 20 common amino acids in biological systems. You can make any number of them in laboratory, but there are 20 common ones in biological systems. All right, so we need at least 20 words to code for those 20 amino acids, right? Because it's unambiguous. There has to be at least one codon for every amino acid. That's what the unambiguous coming down part is. So we need 20 words, right? Well, if we only take 2 letters and we have 4 choices, U, C, A, G, right? We can have UC, UA, UG, we can have CA, or whatever, right? Well, that just turns out to be 4 to the second power, the 2 things. That's only 16 choices, okay? Well, 16 choices is not enough to give you the 20 amino acids plus the term codes, right? So it's not possible that we could have had a coding dictionary with just 2 amino acids, a codon being just 2 letters, right? Okay, so what could we have to have? We'll go to 3. Well, 3 takes you always 64 possibilities, right? 4 and 4 is 16, 4 and 16, 64. That means you've got 64 places in your matrix. You only need 23 of them, right? You need 20 amino acids plus 3 term codes. Okay? So that means you've got all these residual ones that you can reuse for other things. And that's exactly what's happened here, is that you end up with this wobble effect because it takes a minimum of 3 letters to make each codon. If you have 20 amino acids plus term codes, right? So that's just a little bit of simple math. Okay. All right. So now we have our coding dictionary. Now we can go and start talking about the actual process of the translational system. And we're going to do that. We're going to have to do a couple of things. We're going to put tRNAs together and charge them and get the amino acids ready to be moved in. And we're going to talk a little bit about ribosomes, the workbench at which all this is going to happen. Again, I know almost all of this is remedial for us, but this is kind of important to go through it. And we will go through it very quickly as we work through those systems.